My title is Diffusion MRI and the Fiber Orientation Function. My apology for not putting a title out earlier. So first, I'd like to thank Jim and Paul for their support, their labs, and my previous mentor, and the clinical collaborators. So in this talk, I'll focus on the one voxel only, not a whole brain or two voxels, just one. So the first half of the talk is I will introduce the general diffusion imaging. I'll start from the diffusion physics and the imaging techniques. The second half is I'll focus on sources of structural connectivity and the factors that affect the accuracy of fiber orientation functions. So water molecules are not stationary, they move all the time, and the random motion is called Brownian motion. This is an example of the isotropic diffusion. At a small diffusion time, most of the water molecules, they are near the original location. But as the diffusion time increases, some of the water molecules may diffuse at a far distance. And the probability density function of finding the water molecules may look like this and evolve over the diffusion time. If under the biological condition, the molecules may have their favorite direction for diffusion, that is an isotropic diffusion. This is the mathematical expression for the probability of finding one water molecule or one spin at a particular location and diffusion time. The example probability is the integration of the single spin probability over the density function. And the resultant function is called probability density function, which depends on the capital R. R vector is a displacement vector and diffusion time. As we previously said, that the water molecules are not stationary, they move all the time. The evolution of the probability density function over time is governed by this diffusion equation with the diffusion information on the right side of this equation. So the diffusion tensor imaging, one particular solution to this diffusion equation is a Gaussian distribution. And the probability density function is a three-dimensional exponential decay with the diffusion tensor as the covariance matrix. I think I'll skip this. Limitations for DTI. DTI works very well with a single fiber direction and at a lower diffusion weighting, p-values are around 1,000. However, it fails to characterize complex structures. By complex structures, we mean that the angular complexity arising from multiple crossing fibers and the radial complexity arising from the multiple diffusion compartments with the fast and slow diffusivities. If there's a crossing fiber inside the voxel, the diffusion tensor can no longer predict the correct direction of the fiber. And also, since 1999, researchers started to notice that the diffusion signal is not a single exponential decay. The log of the diffusion signal is not a straight line. It starts to bend at a higher diffusion weighting. This is a study of a human brain at the internal capsule. Another study of this red spinal cord. Human study with a normal brain 1 meter, gray matter, and a tumor. So the high angular resolution diffusion imaging, HARD-D, is MR acquisition techniques. It has more diffusion weighting directions than DTI, and sometimes it acquires data at a higher B-value. Although Hardy data can fit into single tensor model, but it actually intends to describe multiple crossing fibers and hope to resolve the angular complexity. The fiber orientation information is described by the diffusion signal profile or by the apparent diffusion coefficient profile by computing the diffusion coefficient at each diffusion weighting gradient direction using logarithm of signal decay, or by the fiber orientation distribution function ODF in a displacement space. The computation method for this, we will discuss it later. These figures shows the directional profiles from the signal and ADC, ODF. So the simulated fiber is along the x-axis with the anatomical color convention, like just Christine described, red for left and right, green for anterior, posterior, and blue for superior, inferior. So with the fiber along the x-axis, this is the signal profile that the directly output from the MRI scanner. This is a planar disk on the YZ plane. This is because the longitudinal diffusivity along x-axis in this case is much faster than perpendicular diffusivity. And the diffusion signal decays much faster longitudinal than perpendicular. Another simulation for two crossing fibers along X and Y. Signal profiles has the highest along the Z direction. The Q-space imaging. So we mentioned that the diffusion signal decay is not a single exponential decay. This study is to measure the maximum B values where the voxels still have a significant signal than noise. As you can see that the gray matter signal are not detectable at a B value larger than 4,000, whereas the white matter still has a significant signals at a B value of 16,000. So why is B value important? Because it affects the diffusion contrast between brain tissues, between different voxels. So in these figures, you can see the contrast between gray matter and white matter goes higher at a higher B value. And it also determines where the diffusion signal comes from. It is hypothesized that the faster diffusion compartment has hindered water from the extracellular space, and the slow diffusion compartment has restricted water from the intracellular space. And the diffusion signal that we measured is a combination of fast and slow compartments. Okay, so if we do the experiment at a lower B value, we can see that the fast diffusion compartment contributes more than the slow diffusion compartment. At a higher B value, here, the slow diffusion compartment contributes more than the fast diffusion compartment. So, only sampling at multiple B values is able to capture the multiple compartments. And the Q-space imaging is an imaging technique that samples at multiple B values with a narrow pulse assumption. The diffusion signal attenuation is actually a Fourier transform with the ensemble probability density function in a displacement space. That means that we can simply do the inverse Fourier transform of the Q-space signals to get a probability density function. And the reciprocal Fourier transform variables are capital R and the Q. The Q vector is linear proportional to the diffusion gradient weighting direction and diffusion gradient duration. I'm sorry that this is not very high contrast. So the B value, in traditional Q-space imaging, we usually use a Q vector to describe the strength of a diffusion weighting. But here in this talk, in this presentation, I'll use a more common term B values. But keep in mind that the relationship with B value and the Q is B is linear proportional to the Q square and the correction term of the diffusion time. So there's two kinds of acquisition scheme for the Q-space imaging. One is multiple B values and multiple diffusion weighting directions. In this method, we can acquire the full Q-space and just use a directly inverse Fourier transform, or we could fit the whole Q-space data into some sort of models. Some people, they do this Q-space imaging using single B value and at multiple directions. Usually, this will be at a little bit higher B value than 1,000. And this is equivalent to the Hardy single shell sampling. This kind of experiment, the data processing has to include some modeling assumption or approximations. These are examples for the full Q-space sampling. The left side is the diffusion spectrum imaging DSI. It has the homogeneous sampling density in the Q-space. It's sampled at a grid point. And the right side is a hybrid diffusion imaging HIDE. It uses a non-uniform sampling strategy with multiple shells. So the data processing, the model base is the famous combined hindered and restricted model of diffusion trumped model, or some assumptions using Hardy acquisition like QBI, parse MRI, or DOT, and the model free. The fiber orientation information described by the isosurface of the probability density function at a fixed displacement distance, or the orientation distribution function. ODF is defined as the radial projection of the PDF. It describes the projected probability of water molecules on a spherical surface. Another way to compute ODF is using Q-ball imaging. It uses the integral over the equator in the Q-space using Hardy acquisition method. And this ODF is an approximation of the original ODF. In addition to these directional measures, Q-space imaging also yields scalar measures from the PDF. For example, the zero displacement probability P0 describes the probability that the water molecules diffuse the minimum within the diffusion time. So the tissue with a high restriction level will have high intensity in this P0 map, like a 1 meter area. And the mean square displacement MSD is the expectation value of displacement squared, and with the Einstein diffusion equation, this is linear proportional to the average diffusivity within a voxel. Also, we could fit the Q-space data into some model. This is the simplest model by exponential fitting with a fast diffusivity and a slow diffusivity compartments. And they are fractional anisotropy. These are corresponding maps. The slow diffusivity maps shows that the 1 meter has a much lower slow diffusivity than gray matter. Also, with the hypothesis that the slow diffusion compartment contains water from intracellular space. So the volume fraction of the slow diffusivity compartment usually is used as axonal density or fiber density. The comparison between the fraction anisotropy and this P0 measure from the rich Q space data is that P0 is more homogeneous to describe the white matter area, whereas the fraction anisotropy suffers from the multiple fiber crossing and may decrease its value to similar to gray matter area. Another example is using the tissue segmentation algorithm. P-value can give you a better white matter segmentation compared to FA. The white matter area some of the white matter voxel is classified into the gray matter. This is one study of this high-D experiment on the shaking pup. Shaking pup is an animal model for the human multiple sclerosis. It's a genetic mutant dog with dysmyelination in the brain. So it's not like directly multiple sclerosis, because multiple sclerosis is demyelination with the brain information and myelin is gone. But the shaking pup is born without myelin, so it doesn't have any inflammation or any confront other issue with the myelin. The upper row is the control dog, and the lower row is for the shaking pup. And this is a histogram for the zero displacement and histogram for the fraction anisotropy. As you can see, for the control dog, the contrast of the P0 between gray matter and white matter is obvious, whereas the contrast of gray matter and white matter is lost in the shaking pup. And fraction anisotropy still do have some contrast, but it's much lower. And this is a longitudinal study. We studied 6 control dogs and 7 shaking pups. And each dog was scanned twice or 3 times at a different age point. Only in P0, MD, and radial diffusivity shows significant change with age. And only in P0 shows significant difference of the slope between control and the shaking pups. Although FA has a difference between control group and the shaking pup group, but it doesn't show any significant change with age or the slope. So DTI is a simple and very elegant approach, but it has limitations. The diffusion angular complexity may be described by using a high angular resolution diffusion imaging approach. The diffusion radial complexity can only be captured with the sampling at the multiple B values. And also, the diffusion signal that we measure is a mixture of a fast and slow compartment, depends on the B value that we select. Also, in addition to the directional information, diffusion imaging can also use scalar measures for white matter biomarkers. So I'll switch gear to the source of structural connectivity. This is a summary of those techniques that use maybe more directions or higher B values and hope to obtain this more sophisticated directional information. And later in white is acquisition at a single B value. Later is in yellow is at the multiple B values. And I also list their preferred B values and the usually number of great diffusion weighting directions. The imaging scan time is linear proportional to the number of directions and also increase with a higher B value. So for example, for the diffusion spectrum imaging with 500 directions, you probably need 2 hours of scan. But why bother? Why we investigate the scan time, the money, to do this complicated experiment? That's because the prevalence of 1 meter voxel with multiple fiber directions is much higher than we expected in the past. In 2007, people reported as a 30% prevalence of 1 meter voxel as more than one fiber. This year, there's a paper published at probably 90% using the constrained spherical deconvolution method. The multiple fiber directions will have impacts on 1 meter tractography. It will cause false negative with early stopping of tracks at this complex area. Also, it could cause a false positive with unreliable fiber direction. You could switch your fiber direction to a wrong direction and create a false fiber track. Also, the diffusion scalar measure, for example, fraction anisotropy, is very difficult to interpret. I mean, we need to interpret with really great caution. And usually in the tractography algorithm, people use fraction anisotropy as an indication of 1 meter. So you may cause the early stopping of these tracks when you using this approach. The sources of structural connectivity may come from the diffusion signal profile directly output from the scanner. Some algorithms work on this to fit model or multiple tensor model or diffusion apparent coefficient profile or orientation distribution function. But the ultimate goal is to estimate the number of crossing fibers and the direction of fibers as accurate as possible at the voxel level. And that is because the angular error is cumulative in tractography, so we want to have a good start in the voxel level. This is just for comparison for diffusion tensor imaging ellipsoids and with the high angular resolution diffusion imaging. At the prefrontal 1 meter area, you can see the single fiber from the corpus callosum and a single fiber track with internal capsule crossing at this area, and also some crossing area from the arcuate fiber and the spheral longitudinal fasciculus. This is a zoom out view of the prefrontal 1 meter area and their fiber orientation function. Here is the ventricle and the corpus callosum go this way. Anterior limb of internal capsule go this way, and there's a crossing here. And here is left to right subarcuate fibers and subcortical arcuate fiber and y direction subcortical arcuate fiber. Here is a singular bundle. Another ODF function, the pond region with transverse pond-type fiber and the medial cerebellum pitangle, cortical spinal tracts. These are the factors that will affect the diffusion signal and then affect the fiber orientation function. So first is the B value. As we previously mentioned, it determines where the signal comes from, or the number of directional the diffusion weighting gradient direction. It directly affects the angular sampling resolution. And different data processing algorithms may create different results. And that is very important, is the imaging noise. This is a simulation of effects of B value on this fiber orientation function. We do these two crossing fibers each fiber has fastened a salt compartment, and the diffusivity is from the in vivo measurement of this paper. And we look at the different intersection angle between these crossing fibers, 90, 75, 60, and 45 degree. At the lower diffusion weighting, the ODF function looks really chubby. As the diffusion weighting increase, the ODF function gets sharper. And the smaller intersection angle between two fibers is able to resolve only at a higher B value. The human data with B value of around 1,000 up to 4,000, and at a higher B value, you can enhance these small fibers here, where you cannot see in the lower B value area. And start to see here, also this one. The number of diffusion gradient directions. The angular resolution for the 50 directions is about 30 degrees, and about 500 directions is about 10 degrees, which means that with this angular resolution, it is impossible to separate two small, like intersection angles smaller than 30 degrees if we use 50 diffusion weighting directions. However, we need to consider the scan time, also the B value that we select. For example, at the lower B value, the ODF looks chubby, so more diffusion weighting directions doesn't help that much. And also, it depends on the computation algorithm you want to use. More complex model requires more sample numbers. So then you will need more number, or sometimes you need multiple B values. The comparison between ODF using QBI 50 directions versus 92 directions, and more direction is able to resolve a smaller intersection angle. The human data with 252 direction versus 162 direction, more direction is able to show these two fibers more clearly than a smaller number. And also here. Different computation algorithm is here compare 4 kinds, and some computation algorithm is more sensitive to this smaller intersection angle compared to others. And also using ODF, if we use the original radio projection ODF, the profile looks chubby, but if you use the QBI ODF, is sharper. And the space where we compute this ODF function, for example, in the displacement space, you got this wide waist, and if we compute ODF in the Q space, it's narrow. Also, there's some processing regulation. If we use a high-pass filter in this spherical surfaces, like using spherical harmonic decomposition reconstruction to get rid of a higher order part, and which usually decrease the noise, and you can get a more clear crossing fiber. Signal-to-noise ratio, it affects the estimation of a number of fibers, because sometimes the noise create false peaks within a voxel, so that it could cause a false positive in tractography. Also, the noise tends to broaden the ODF profile and increase the uncertainty in tractography. I also list these MRI parameters that could affect the image signal-to-noise ratio. This is a one trial simulation of noise at a different intersection angle, and different signal-to-noise level. This is the results of 100 trials. The left side is noise-free original 2 fibers with different crossing intersection angle, and I compared the different methods. But nevertheless, the original number of fiber is 2. So at a larger crossing intersection angle, most of the methods do pretty well. They can predict 2. But the smaller signal-to-noise ratio has a higher standard deviation. At a smaller intersection angle, all of them is not. Only predict 1 fiber rather than 2. But the intermittent intersection angle, some method could predict good like 2 fibers, and also larger noise will tend to underestimate the number of fibers with higher standard deviation. Last is the noise effects to the angular arrow. This is a simulation of a single fiber, and I rotate at 100 directions. At each direction, I simulate 10 trials at each signal-to-noise ratio. And the angular arrow is defined as the I originally simulated direction and the direction that I predict from the ODF profile using very simple maximum likelihood algorithm. And then I average over 100 directions and 10 trials. So at a lower signal-to-noise ratio, we will have a higher angular error with a larger standard deviation for all methods, but some methods tend to do better than other algorithms. Summary. So more sophisticated directional information than diffusion tensor ellipsoids is needed for more accurate fiber stratigraphy. The angular error is cumulative in 1 meter stratigraphy, and it propagates from seeds to destinations. And different computation algorithms or different diffusion imaging techniques has their strengths and their limitation. Also, these different techniques may have different sensitivity to noise. They have their preferred B values and a number of gradient directions, which could affect their accuracy in estimating the fiber numbers. So last is guide definitely has thousands of crossing fibers. And thank you for your attention.
So you have this 2 exponential model to describe the value of the signal you obtain, right?
Yes.
I wonder if you try 3 exponential model. Let me say why am I asking. In previous talk, Yu-Chien wonderfully drawn that water molecule which actually changes the compartments from time to time, right? And that's one of the diagnostics for problems with myelination where they try to escape, right? And in the DTI model, we have increased orthogonal component, right? But is there a direct measure to describe how much of this diffusivity which crosses the membranes is? And I guess with 3 exponential model, you could have hoped that kind of the slowest one would be the one which we are after.
Right. The 2 by exponential model is the simplest model I did. But of course, other people, they do multiple model to describe the restricted diffusion, the stored diffusion compartment part with different orientation, even with different diffusivity. So.
But in your shaken pop especially model, right? That's, biological model, right? So that probably would be a good data word to try this multiple exponential model to see if there is clear indication of much slower diffusivity components.
True, that's right. Thank you.
All right.
In Christian's talk, he showed that myelination didn't have much of an effect on fractional anisotropy, which I think was echoed in your study with the shaking pups, that fractional anisotropy was not affected, which did not show a difference between the shaking pups and the control animals. But the P0 did show a difference. So can you just be more precise about what is P0, why is P0 sensitive to myelination, whereas fractional anisotropy is not?
One explanation could be that when there's a myelin, the tissue is packed, so the free space for the water to diffuse is limited. So it's more restriction of the water molecules, so that the zero displacement probability increase. And I wouldn't say that P0 is a direct biomarker to myelin, but it reflects the restriction level of the tissue or the microstructure. And in any case, the restriction level increase, P0 increase.
Fractional anisotropy is used also as a measure of sort of the integrity of a white matter tract, and it changes with things like training. So if someone learns a new task, the white matter tract that's critical for that task shows an increase in fractional anisotropy. Would P0 show the same kind of effects that suggest whether or not the fibers within a voxel are well organized or not?
I would expect P0 will change and even has higher sensitive to this fractional anisotropy measure, but we need to do the experiment to show that.
I just wanted to comment on just the one thing is that there's been a bunch of shiverer mice studies as well that, so it's not true that the FA doesn't change. It just doesn't change by as much as you might expect when you get rid of all the myelin. So it still changes anywhere from 8 to 20%. But considering that you've wiped out all the myelin, you might have expected that you'd have like dramatic changes. And the reason you don't is because you still have all the axons that are intact and you have all the axonal membranes. The problem with all these models is that it also assumes that the axon packing is the same, the axon diameters are all the same, and so on between the two models, which may not be true. So if that's not the case, then you still have all these membrane structures restricting the diffusion perpendicular and still causing quite elevated FA. That's why you see it in the non-myelinated olfactory nerve of the garfish. It has a high FA because of these small little axons, but you still do see a modulation of FA with demyelination. And there's also been a nice study where people remyelinated and you see increases of FA in an animal model where they put in some precursor stem cells, and the area that they got more myelin also increased in FA. So it's not completely true that it's just not the only story.
So let me make sure that I've got this straight. So in these animal models, there's lack of myelination. Are the fibers packed more tightly? So is there an increase in the number of cellular membranes because there's less myelin taking up space? Is that true?
I think you'd have to look at, I can't recall the papers, but I'm pretty sure that that's probably the case. I mean, you don't just have big empty spaces, I think, around them. The myelin's gone, and then you have this big gap between axons. I think if you did that, then you would see much markedly different changes in your FA parameters and the other parameters as well, but I don't think that's the case.
In the beginning of your talk, you showed images of different p-values,
Right.
all the way to almost 10,000. And I'm really impressed by the quality of those high p-values. Could you say a few words how you acquired the data?
I acquired the data at the 3 orthogonal directions and like multiple p-value steps, like 16, and I repeated like more than 10 times, and then average.
Okay, so 10 times
Yes.
3 and then average.
Yes.
Okay, thank you.
And it wouldn't take too much time because it's just 3 directions.
No, I'm just impressed by the data. It looks very nice.
Thank you.